These combined methods can answer key questions in the field of epididymal research, such as what protein targets are involved in the regulation of sperm maturation and storage. The main advantage of the epididymal isolation technique is that it is also applicable to isolate a similar population from other regions of the epididymis among different species.
To begin this procedure, transfer the epididymal paraffin sections to a fume hood. Add enough xylene to the slide jar to completely immerse the tissue section. Rehydrate the tissue sections by immersing them in grated ethanol solutions diluted in purified water, as outlined in the text protocol. Wash the sections in a slide jar for 5 minutes with sufficient PBS to completely immerse the entire tissue section. Next, decant the appropriate antigen retrieval solution into a slide rack. Microwave the solution until it is boiling. Completely immerse the slides in this solution, subjecting the tissue sections to heat-induced antigen retrieval solutions. After this, remove the slide container from the microwave and let it cool to room temperature. Rinse the slides in a jar containing PBS for 5 minutes. Using a liquid repellent slide marker pen, trace around the tissue section. Transfer the slides to a humidified container. Apply blocking solution for 1 hour at 37 degrees Celsius. Then, rinse the slides once with PBS. Incubate the sections with an appropriate primary antibody diluted to an experimentally optimized concentration in filtered